Hello, we will continue our discussion on prostate cancer tissue imaging. We have started seeing different tissue images of prostate cancer in terms of different molecules that are there, and I showed you one example that is myosin 11, which is cytoskeletal protein, and that was spread across the tissue. Now, I will talk about those three proteins that we were left with out of comparison between the imaging that we got for different peptides and analysis characterization of different proteins that we got in a simultaneous proteomics approach. So, we talked about 52 proteins that we were seen. And then we ruled out 41 because of not very different distributions, clear distributions across the tissues. Then we removed 8 which are uniformly distributed, and out of which one example I showed you. And then we are left with 3 interesting ones which are perhaps related to the prostate cancer. So, here the figure represents the distribution of a tryptic fragment 1175. 1175 is a tryptic fragment across the prostate cancer tissue section. The tissue proteomics results showed that the observed tryptic fragment is a part of protein viculin consisting the residue from 237 to 246. So, this is basically the MALDI image of that 1175.6, that is the peptide originated from viculin, and that peptide image basically interprets or basically reflects how viculin, the protein, is distributed across the tissue. Now, figure A, this one as usual, it is H&E stained tissues where encircled portions where viculin was overexpressed. And as in MALDI image of serial section indicating the same, because here it is most intense in the heat map, yellow supposed to be most intense and next to that is red, and in this portion the distribution of your intensity of 1175 is the highest one. So, the encircled portion represents the region where the intensity of the peptide was observed to be higher compared to the rest part of the tissue. Now, this represents the isotopic distribution, figure C. So, isotopic distribution of 1175.6 in the mass spectra is shown in figure C, where this is obviously monoisotopic peak. The second one is 1C13, third one is 2C13, fourth one is 3C13, and so on and so forth. Okay. Now, figure D, this represents 20x expanded view of a small representative part, this tiny representative part within the encircled region of H&E stained tissue section that showed viculin being overexpressed in the region, in that region, which is basically having dense population of cancerous epithelial cells that might have undergone clonal expansion. A process of acquiring clones with variable type of cancer associated genetic mutation in addition to the initial genetic mutation which is developed the cancer. So, that is what is called clonal expansion. And it would potentially be presented with increased aggressiveness of the cancer. So, this is the region, that is where it is shown, or some small portion is blown up, and you can see that how the cancer cells are so closely, densely packed here. Now, viculin is coded by the VCL gene, a cancer genome atlas TCGA, reports shows that the viculin coded by the gene VCL, so that gene was altered and corresponding mRNA was overexpressed in 4% patients of the patients screened for prostate cancer. So, that is from a different study. Now, vinculin is a cytoskeletal protein localized in the cytoplasmic phase of the junction between cells and extracellular matrix. It binds another cytoskeletal protein, taline, and potentially links the integrin proteins of the cell membranes to the extracellular matrix. So, it has been reported that the presence of vinculin in focal adhesion, that is protein assemblies forming the site of contact of cells with the extracellular matrix, that is critical, focal adhesion, that is critical for integrin mediated cell migration. Previously, immunohistochemical studies on tissues with amplified VCL gene expression from patients with castration resistant prostate cancer revealed overexpression of vinculin, and it was reported that the vinculin is a major driving gene of 10q22 amplification in the prostate cancer. So, whatever we are finding is nothing new, but it is very well correlating. That is the most important part. So, thus overexpression of vinculin might be associated with enhanced tumor cell proliferation in prostate cancer pathogenesis. This data showing that the overexpression of vinculin by cancer epithelial cells of the prostate tissue is in agreement with the established role of this protein in prostate cancer. So, at least one good correlation, good image we observed for in our MALDI imaging study of prostate cancer tissue. Let us move to the second one. Another peptide 1143. So, it must be again originating from a different protein. So, figure represents the distribution of a tryptic peptide. Remember, we have used trypsin as a proteolytic enzyme. So, tryptic peptide with MYZ 1143.6 on the prostate cancer tissue section. Now, proteomics data, which obviously should be used, must be used to characterize the protein. So, that data revealed that the peptide fragment is the protein ribonuclease T2 comprising residue from 144 to 153, that stretch. Okay. Now, figure A again corresponding to a region in the H&E stained tissue section, and that is encircled here, where ribonuclease T2 was overexpressed in the MALDI image. That is what we have been seeing through the image, mass spectrometry based MALDI image of ribonuclease T2, that is depicted in figure B. Now, the encircled part represents the region where the intensity of the peptide was observed to be higher compared to the rest part of the tissue. Okay. The isotopic distribution of the peaks 1143 is shown here as C. So, 1143, then this is monoisotopic peak, this is peak with one C12 being replaced by C13 and so on. There may be some overlap from other peptide, which is why the third signal seems to be little more than the second one, or almost equal, but it is by actually naturally it should be less. So, something else has contributed and the spectra picked up both together. So, that intensity got summed up here. Anyway, so that is a separate issue, but this is the peptide and its isotopically distributed peaks are also present. So, this is a real one, not the noise. Okay. Now, figure D represents the 20x expanded view of small representative part of the encircled region of the H&E stained tissue section illustrated in the cellular population in this region. Okay. So, upon correlation with the histopathological image, the MALDI image distribution of ribonuclease T2 across the tissue section, so that it was overexpressed in the region infiltrated with immune cells such as macrophages and neutrophils. Okay. I am repeating again, it is very important statement. Upon correlation with the histopathological images, the MALDI image of distribution of ribonuclease T2, which is what is shown here, across the tissue section showed that it was overexpressed in a region infiltrated with immune cells such as macrophages and neutrophils. Okay. Now, ribonuclease T2 coded by a human gene RNA-set2 that is secreted by epithelial cell into the extracellular milieu. Now, TCGA, what is TCGA saying? TCGA database reports that an alteration of RNA-set2 gene and a subsequent mRNA overexpression in 4% of patients with prostate cancer. So, that is again known. Now, the gene was classified as belonging to the category of tumor antagonizing gene or malignancy suppressing gene. Now, TGA or MSG, meaning tumor antagonizing gene, TAG, or malignancy suppressing gene, MSG, the class of genes are known to code proteins which are required to mediate cellular response in the tumor microenvironment to prevent the process that leads to malignancy, such as angiogenesis. So, it has been reported that the overexpression of RNA-set2 gene results in a trigger response from immune cell such as monocytes and macrophages, indicating a biological mechanism based on microenvironmental control to prevent the growth of tumor. So, that means this immune system itself, when it is trying to kill the cancer cells, so that event is very much associated with the overexpression of ribonuclease T2, which is what perhaps we are seeing here. Now, in addition, the activity of protein antigen target A kinase anchor protein AKP4 involved in several malignant properties of tumor. What are those? Such as arresting cell cycle, preventing apoptosis in several cancers like prostate cancer, esophageal cancer, multiple myeloma, and is actively suppressed by the ribonuclease T2. Okay. So, what is it saying? The activity of protein antigen target A kinase anchor protein, that is AKAP4, involved in several malignant properties of tumor, which are those like arresting cell cycle, preventing apoptosis in several cancers like prostate cancer, esophageal cancer, multiple myeloma, and that is actively suppressed by the ribonuclease T2. Okay, because if ribonuclease T2 can suppress this, then cancer cell, meaning this preventing of apoptosis can happen. Okay, meaning apoptosis by default is a process which cancer cell doesn't want to, and then ribonuclease T2 will act against that. Okay. So, ampiregulin is another protein involved in regulation of cancer cell invasiveness and is reported to be negatively regulated by ribonuclease T2. So, no matter whatever literature informations that we are seeing in terms of activity of ribonuclease T2, it is always just to negatively regulate a protein associated with cancer or some way or the other to prevent cancer by and large. So, then it is very well again correlated with its expression that we are seeing through MALDI image and the literature information that is talking about its functional property. Therefore, in this study, the observation on overexpressed ribonuclease T2 in a region infiltrated with macrophages and neutrophils corroborated well with the literature information available for this tumor suppressor protein. So, one good correlation one meaningful image that we got from MALDI mass spectrometry imaging of prostate cancer. Let us see the third candidate. So, that another peptide, another tryptic peptide 2295.2 on the prostate cancer tissue section, that actually seems to be a fragment of a 60 kilodalton protein, which is heat shock protein, comprising residue that is from 371 to 389. So, that is what the proteomics data is showing. Now, this figure, figure A again shows the corresponding region in H&E stained tissue section, that is the encircled portion where protein 60 kilodalton is overexpressed in the MALDI image. This is the MALDI image of the serial section of the tissue, and this portion is actually it's here, and this basically shows where the protein heat shock protein 60 kilodalton that is overexpressed here, and this is how H&E stained tissue section looks like. Now, the encircled part represents the region where the peptide was observed to be higher in expression compared to the rest part of the tissues, and the isotopic distribution of the peptide having M by Z 2295.2 in the mass spectra is shown in figure C. So, obviously 2295.2, this is monoisotopic peak, this is one C12 being replaced by one C13, 2 C12 replaced by 2 C13, 3 by 3 like that, and naturally this is having more than 100 carbon atoms. So, naturally we do expect 1 C13 by default would be there, which is why the monoisotopic peak is not the most intense one, but the second one is the intense one. That we have studied previously also. I am just mentioning that everywhere, no matter the peak comes from tissue or from solution phase analysis, from toxin, from any organism, the natural scenario is same. Okay. Now, let us talk about some function and associated correlation of this 60 kilodalton heat shock protein with cancer, specifically prostate cancer. So, it was observed that 60 kilodalton heat shock protein got overexpressed in the region of the tissues having cancerous epithelial cells surrounded by the visible amount of stroma containing immune cells. And this 60 kilodalton heat shock protein is coded by gene HSP60 and belongs to the family of chaperone. So, it is present in the cytosol, cell surface and extracellular space and in the peripheral blood. However, it is mostly abundantly located in the eukaryotic cell mitochondria and is induced in response to stresses such as heat shock, which is the name it coined for heat shock protein. So, it has important function in transport and folding mitochondrial proteins. This protein is also reported to play a role in the transformation, promotion of angiogenesis, which is related to the cancer and metastasis of the cancer cells. So, what is literature is saying? According to TCGA database and the report, that mRNA of 60 kilodalton heat shock protein gene is overexpressed in 7% of the patients with prostate cancer. For other two proteins, we have seen that it is 4%, and here in this case it is 7%. An increased expression of the gene has been reported in multiple cancers such as glioblastoma, colorectal cancer, gastric cancer, breast cancer, ovarian cancer, liver cancer, bladder cancer and head and neck cancer, a whole host of cancers. It is present in the present study. This 60 kilodalton heat shock protein was found to be localized in different regions of the tissues that have well organized pattern of cancerous epithelial cell, that is while describing the H&E stained tissues, I showed that to you. Now, what it indicates, this might indicate that 60 kilodalton heat shock protein was secreted in the extracellular region by the cancerous epithelial cell of the prostate gland. Therefore, the observation of this specific distribution of 60 kilodalton protein, heat shock protein in prostate cancer tissues corroborated well with the reported literature information about this protein. So, at least 3 protein images which I showed you, their presence in two-dimensional heterogeneous distribution across the tissues corroborated well with the reported literature. Now, I will show you how these images are seen in needle biopsies. So, I will show you the needle biopsies. These are all MALDI-image distribution of 3 different peptides that we have described. 1175, that is from viculin, 1143 ribonuclease T2, and 2295 heat shock protein, HSP60. So, how they are distributed from needle biopsies isolated from different patients, 4 different patient samples. So, these are needle biopsies. This is actually different portions that is encircled here, how those are appearing in the under microscope in H&E stained tissue section, and then how the image, MALDI-image of this entire needle biopsy tissue and the different encircled portions, how it looks in terms of expression of 3 different proteins that we have discussed, that is shown here. So, this is from a patient 73 years old, this is 63 years old, 67 years old, 67 years old, another patient 70-80 years old, another patients, all are cancer patients. So, MALDI-image of the distribution of 3 peptides with M/Z 1175.6 of viculin, 1143.6 of ribonuclease T2 and 2295.2 of 60 kilodalton heat shock protein in 4 representative needle biopsies collected from patients diagnosed with prostate cancer are shown here. Okay, so if molecule is there, then it will be there forever, meaning irrespective of the patient, irrespective of the way that you see. So, once we got to see these 3 molecules that are very much related to the cancer, prostate cancer, that's what we have discussed in today's class, that is seen in the needle biopsies also. Now, I will end the class by talking about proteomics finding, which we have discussed, but we were talking about identification and characterization. Now, I will be talking about quantitative proteomics, quantitative tissue proteomics. So, this is the beautiful chromatogram that is obtained from nano-LC, which represents different peptides that have come out, nano-LC ESI-MS that was used to characterize protein, identify it and to quantify it. So, the quantitative proteomics data of 7 needle core biopsies collected from patients with prostate cancer, 10 tissues core obtained from patients with BPH were compared, and ribonuclease T2 and 60 kilodalton heat shock protein were observed to be overexpressed by 22.26-fold and 6-fold respectively in the prostate cancer tissue. That is what this table shows. Ribonuclease T2, this is control, normalized and averaged samples and the fold changes. I told you that here control means BPH, because without any problem in the prostate, no ethical committee will allow you to take the prostate tissue. So, we do not have absolute control in that sense in the entire study. We have the BPH, that is non-cancer stage, it is not cancerous, so that is our control, and prostate cancer is the sample. So, whatever fold change that we are talking about is the comparison between the BPH and the cancer. Okay. Yes. Now, however, compared to BPH sample, there was no significant change that was observed for viculin. which we were talking about, we have seen that it is very much associated with the distribution that corroborates well with the histopathological distribution that we see under microscope of cancer cell, but in proteomics we didn't able to see a significant overexpression in case of cancer. There is a reason that we will discuss about it, most likely though, because that's where some discordance is there. So proteomics data was normalized. How did you normalize? By dividing amount of each protein by the summation of amount of all proteins present in the sample. The experimental setup to perform tissue proteomics includes homogenization of the tissues, which results in loss of heterogeneous spatial distribution of the proteins across the tissue section that we have been talking right from beginning. That's the main advantage of doing tissue imaging. And then disappearance of the concentration gradient of the marker of pathologically important portions of the experimental tissue section with respect to total content. So all we are saying that when we are classing the tissues, we are not actually segregating the portions or taking out the portion which is been filled with cancerous cells. A tissue is having cancer cells as well as normal cells. When we are classing it, then if the normal cells are more, then as we are losing heterogeneous distribution, so it's no more heterogeneous, is homogeneous. So then the gradient of that particular cell which was for the protein which was more in cancer cell, that may be diluted because it's getting now mixed with large amount of normal cell isolated from same tissues. So this might be the reason why differential level of vinculin was observed in, meaning no significant differential expression, I should write. Please read that, it's interpreting in a different way. Actually, vinculin was not significantly differentially expressed here, and the reason for this could be this loss of concentration gradient when we actually cross the tissue, and that perhaps led to disappearance of in terms of fold change in the case of cancer that we are not seeing for vinculin. So that was observed in the proteomics, whereas it was observed specifically located in small area in the MALDI imaging tissues having dense population of cancer cells. That's what I showed you. So whenever the cancer cell distribution is dense, vinculin is very much there, but that was a part of the tissues and rest part was normal. So or there was some portion normal as well in the tissue. So that normal portion is making it diluted so that it doesn't show up as overexpressed one. So with that, I'll end the prostate cancer tissue imaging recorded through MALDI. In the next class, I'll describe about this prostate cancer tissue, maybe different tissue because it's from completely different study, but the disease is same, prostate cancer. How does image look in terms of distribution of small molecule? Here we are describing proteins, there we will see how it is small molecule like metabolites are seen in the imaging platform, and then we will use different strategy, desorption electrospray ionization based mass spectrometry to image the tissue. So I'll end the class here. Thank you.